Mitochondria resemble bacteria via endosymbiosis. 2. Mitochondria found in eukaryotic cells. 3. DNA double-stranded, RNA single-stranded. 4. DNA replication occurs during S phase. 5. ORC initiate, MCM unwinds DNA. 6. DNA polymerase needs primer, RNA doesn't. 8. Amphensin-proved proteins refold spontaneously. 9. Garrod-linked genes to enzymes. Alkaptonuria. 10. Avery identified DNA as transforming principle. Streptococcus. 11. Hershey-Chase confirmed DNA stores information. 12. Ligand-gated channels respond to signals. 13. Mutant grows with B and C. 14. Griffith showed DNA transformation ability. S dead. R live. S live. R/S dead. Disulfide isomerase reshapes protein bonds. 2. RES anchored via lipid tail. 3. Tyrosine kinase adds phosphate groups. 4. Intracellular receptors bind hydrophobic signals. 5. cAMP kinase activated by cAMP. 6. Ubiquitin tags proteins for degradation. 7. Insulin synthesized processed in pancreas. 8. Epinephrine increases blood glucose levels. 9. Allosteric inhibition changes enzyme shape. 10. Lactoperon regulated by lactose. 11. Low GTP halts elongation process. 12. RAS mutations drive cancer growth. 13. Glucagon increases glycogen breakdown rate. 14. Cell signaling involves reception, transduction, response. 15. Paracrine local, endocrine distant, autocrine self. 16. Ligand binds receptor, initiates signaling. 17. Aldosterone produced by adrenal gland. 18. Nitric oxide relaxes blood vessels. Fatty acids create cell boundaries. 2. Phosphoryl group identified in nucleotide. 3. Glutamic acid forms ionic bonds. 4. Ribose synthesizes nucleotide building blocks. 5. Serine targets glycosylation modification. 6. Proteins or polypeptides ionize chains. 7. DNA strands held by hydrogen bonds. 8. Basic polar, nonpolar amino acids. 9. Histones condense and organize DNA. 10. Griffith showed bacterial transformation occurs. 11. Avery proved DNA is transforming material. 12. Hershey-Chase used radio-labeled 32P. 13. Higher GC increases DNA melting temperature. 14. DNA's complete information is genome. 15. DNA templates RNA production directly. 16. Replication terminators unnecessary in eukaryotes. 17. Organelles originated through endosymbiosis. 18. Mitochondria share DNA, ribosomes division. 19. Miller's experiment formed amino acids. 20. RNA catalyzes nucleotide polymerization. 21. Antibodies locate specific cellular proteins. 22. E. coli small, fast mutable. 23. Hydrothermal vents supported Luca's origin. 24. Water most abundant cellular molecule. 25. Cell membranes primarily phospholipids-based. 26. DNA pyrimidines or cytosine, thymine. 27. Proteins linked by peptide bonds. 28. Viral genome, likely single-stranded RNA. 29. Protein structure determined by sequence. 30. Membranes cross gradients via active transport. 31. Membrane proteins anchored by prenyl groups. 32. Proteins move laterally in membrane. 33. Membrane functions depend on proteins. 34. Glycolipids only in outer leaflet. 35. Tight junctions organize glucose transporters. 36. DNA strands antiparallel, directional sequences. 37. High K plus inside, not plus outside. 38. Neurotransmitters open ligand-gated channels. 39. tRNA synthetases link amino acids. 40. mRNAs read 5 feet minus 3 inches, synthesize NC. Breakage/reformation enzyme. Protein disulfide isomerase. Endoplasmic reticulum. Polyribosomes. Multiple ribosomes translating mRNA simultaneously. Protein secretion direction. Signal peptide targets rough ER. Chaperone's role. Assist protein folding, prevent aggregation. Phospholipid synthesis. Occurs flipped by flipases. RES insertion. Lipid-modified anchors intermembrane. Secretory pathway. Golgi regions, vesicular transport. Polycistronic mRNA. Encodes multiple proteins, prokaryotes only. Protein kinases/phosphatases function. Add, remove phosphates, regulate activity. Allosteric regulation. Molecule changes protein shape. GTP/GDP in YEF2. Activates/inactivates elongation factor protein synthesis. RES cancer mutation. Always active, uncontrolled cell division.